All right, everyone, now that we know all of the parts of a neuron, we should talk about what those parts do. I'm assuming you know all the parts of the neuron. If you don't know the parts of the neuron, go back an extra couple of videos or a couple of chapters. But in this one, we are going to review that a little. So let's start with a soma, or a cell body. So the job of that is just to kind of hold all of the organelles. It holds the DNA inside of a nucleus. I say that's just what it does. I mean, it does actually quite a bit. It will make the neurotransmitters, which are the chemical messages we can release at the end. So it's kind of like your factory part of your cell. Off of that soma, and I guess I could label that, we're going to have branches that will receive information. So those branches up here are going to be our dendrites. Dendrites are typically very branched. That branching can change through life. These neurons are so cool. They adapt to our learning, our environment. And if there are cells that you are using a bunch, they will start to branch a lot. And there's a lot of cool things that we can talk about with learning and exercise and enriched environments and how it helps these branch even more. So the job of a dendrite is to receive information. So these are going to be your receptors. And then at the end, you're going to pool a bunch of information together and you'll kind of funnel it into an axon. And the axon is where you send your official message. And at the end of that axon, you'll typically see some branching, and at the end of the branches, we'll have a place where it gets a little bit wider because we're storing chemical messages in there. So we have our axon terminals down here where you have the branching, and then we have the synaptic knob. And inside of that synaptic knob, we'll have vesicles that have neurotransmitters, and that's what those little circles are. And so hopefully you remember some of that basic anatomy. Now let's talk about the phys. What is actually happening here when I say we're receiving information? So let's assume then that this is a sensory neuron. To be a sensory neuron, what you're going to do is you're going to pick up information from your environment that's brand new to you. It's not anything you're already aware of. It's not something your brain has already processed. This is something brand new from the environment that we have to convert into an electrical signal. The way we do that is we have ion channels that open in response to different stimuli. And these are gated ion channels. And gated ion channels will open in response to different things. So for example, it could be a physical push. That's a mechanically gated ion channel. And we call those mechanoreceptors. So if we're looking at, say, a physical touch, what will happen when you have a physical touch is that the cell membrane will have this channel here that normally maybe it's touching. But then if I push down here, it will distort that membrane, and then it will create a little gap. Because now we have a little indent, right? For like, say, there's my finger. That's a drawing of a finger. And it made a little gap, and now we can get a flow of charges. And if that flow of charges is something positive like sodium, we're creating a flow of positive charges. And overall in the big picture, that is the signal to flow of positive charges. Because remember, the basic setup for most of the cells in your body is that the inside of that cell is going to be negatively charged compared to the outside, because you have large proteins in here. We also have some potassium in here, so there are some positive things, but outside of here we have a lot of sodium and calcium. We'll come back to that later. And so if we want to create a signal, we actually have to turn the inside from -70, that's what they're usually at rest, up to threshold. And that's a little bit higher, that's at -55-ish, depending on the cell. And so we're going to get a flow of charges. We're going to let sodium in by just pushing the membrane, and we can get closer to threshold. We could use a chemically graded ion channel. So we call those chemoreceptors. And so again, here's our membrane, here's our ion channel that maybe is closed, but it has a little groove on its surface for something to attach to it. And that something might be a chemical that's in our air, that's an odorant that might hit the top of the receptors on the top of the nose. It might be a chemical that's in our food that'll touch those on the taste buds. And so we can respond to chemicals. We could respond to light, those would be photoreceptors. We could respond to pain, those are nociceptors. I'm getting a little ahead of things that we didn't do in the chapter, but hopefully you get the idea. There are different types of receptors. There are also voltage ones, but voltage wouldn't be if it's brand new from the environment, because that's electrical. You shouldn't be getting electrocuted. So here on the dendrites, we'll typically have these kind of receptors. We can also have a thermoreceptor responding to temperature. And so what you're getting is a flow of charges. You're letting sodium in, you're creating a flow of positive things, and this is what we call a graded, oh my gosh, words, gated ion channels opening create a graded potential. So gated ion channels up in the dendrites create our graded potentials. And so a graded potential, my marker's drying out on me, is any change from resting. So graded potential, any change that happens up here. Dendrites, somas, they're receiving information, you get flow of charges. Graded potentials, these small little charges, so let's say we get a bunch of them coming in. So we have a bunch of sodium coming in. They're going to work their way in this direction. And right here is the location of what we call summation. So this is the axon hillock. And that's where summation occurs. That's where you add up these graded potential, these small changes. So in the case of this sensory neuron, we're getting these small changes. So maybe you're opening a few of these chemically gated ion channels and you're letting some sodium come in. If there's enough sodium there, if you get to threshold, that's your key. If you can get to threshold, you send an action potential. So that is the amount of positive sodiums that you have to get to say, yes, this is important to us, we need to send that information along. There are two different types of summation. You could do spatial summation, or you could do temporal. Temporal is about timing. So let's say instead of a whole bunch of mechanically gated ion channels getting opened at the same time, maybe it's just one that just keeps constantly getting over because it's like a constant stimulus. Think temporal, think timing. So you're getting a lot of them over and over and over again, open, 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 you get a lot of sodiums in that way. Spatial summation would be more about kind of utilizing different spaces. So different ion channels over here, maybe some over here, and they all open at the same time, and then you get a flow of sodium. So there are two different ways we can add up these positives to get to that negative 55. If you get to the negative 55, you are going to create an action potential. All action potentials are the exact same size. So if we look at an action potential, if we decide to create one, why it's different here is because these type of ion channels, those were out here on the dendrites, they're responding to something new. The ones here are voltage gated. So voltage gated meaning that they only open when you get to a certain charge. And so voltage gated, responding when you get to threshold. So you have to go from negative 70. So if we were to draw this out, kind of like a voltmeter. So we take and we measure the charge. So we'll have this in millivolts here. We'll have time down here. And let's say we're hanging out here resting at our negative 70 millivolts. And then we start to get small changes, small changes. We don't do anything about it until we hit this negative 55. And so we have small changes. You might even go down, you might come back up, but once you hit here, now we're gonna open these voltage-gated ion channels. Those voltage-gated ion channels are sodium ion channels. And if they open, because of your chemical and electrical gradient, those sodiums are gonna go in. That is going to cause what we call depolarization. You're losing your differences. Initially, remember, you're negative on the inside. Think of your outside as like your 0. So you're negative 70. That's a big difference. When you let positives in, you're losing those differences. And so you're depolarizing. So depolarization happens here. So depolarization, sodium's coming in. Once you get to the peak, which is at positive 30, those sodium ion channels will close, because again, it's all voltage gated, so they're responding to different charges. So at the positive 30, the sodium ion channels then close. We now need to go back to the negative charge. The fastest way to go back is actually not to push the sodium out, because remember, there's still a lot of sodium out here. The sodium doesn't want to go where there's already some. Things like to follow their own gradient. So the fastest way to get it to come back down is actually opening potassium, because then potassium channels are going to allow potassium to leave. And when I say in and out, I mean in and out of the cell. I'm going a little shorthand here. So potassium going out allows us to reestablish, so we're going to call that repolarize. So you're going to reestablish that negative charge. Usually what will happen is you go a little too far, because when you get back to your negative 70, you'll drop a little bit too low. We call that hyperpolarize because we overshoot it. And then we'll eventually come back up because we'll use the sodium-potassium exchange pump. Remember that one? We're kind of flipping them. So we're going to push the sodium out and we're going to pull the potassium back in to get us back to what it should look like at rest. So that's the action potential. So that's what this whole thing is right here that I was trying to show you. So we create a signal by getting to threshold, adding up those graded potentials. Graded potentials exist because you open gated ion channels using a stimulus. So stimulus opens a gated ion channel. You get a flow of charges. Those charges are added up at the axon hillock. If you get to threshold, you can create an action potential. The action potential is what travels down the axon. There are different ways it can travel, depending on if you are insulated or not. So we can go down that axon, we get to the synaptic knob. Once you get to the synaptic knob, the job there is to release the neurotransmitter. So what will happen, where do I want to stick this? Let's color code a little bit since we're pink over there. So we get down to that synaptic knob, a little squeaky. We have chemical messengers in there, and then there's a space, and on the other side there are receptors for those specific molecules. So say if that's acetylcholine, these would be called acetylcholine receptors. When your action potential comes down, right on this membrane is a chemical or a voltage gated. So the action potential will stimulate them to open. So action potential is going to stimulate voltage gated ion channels to open. This lets in a new molecule. This is calcium. That's why calcium is so important for our body. Our bones will give it up regularly if we don't have enough in blood. Calcium comes in and triggers the release from this vesicle. So it'll fuse with the membrane. So I think of it kind of like the cab driver for this little bus. Then those messages can be dumped out here into this space where then it can bind to this cell. And then this is another chemically gated ion channel. And we can let in positive things or negative things depending on what kind of message we're trying to send. So let's kind of break that part a little bit more. So this is the synaptic knob. So the synaptic knob is releasing the neurotransmitters. So that's what these little dots are, neurotransmitters, NT for short. The space here is the synaptic clef. This over here on this side, that's a receptor. And that is a chemically gated ion channel. Now here's what's cool. The type of charge it lets through is dictated by what kind of receptor you're using. So if that neurotransmitter is released here, you could be trying to tell the cell, hey, we want you to get to threshold. Remember, to get to threshold, you're going to make a cell more positive. And if that's the case, then we could have those positive sodiums coming in. So we could say, yep, we want you to get to threshold. We're going to let sodium in so we can become more positive. And so if we become more positive, that gets us to threshold. So we're going to get closer to threshold. This is what we would consider, and again, it's a graded potential, but a specific one because we're postsynaptic. We're after a synapse. So a neuron's talking to us. This would be considered an excitatory signal. So we have an excitatory postsynaptic potential. We're saying get closer to threshold. A neuron is telling the next neuron, hey, I want you to do something. But you could sometimes say, you know what, I don't want you to do anything. Whatever this message is that we got from the environment, we want to say, oh, don't respond to that. Don't do something. We want to inhibit this. And so in that case, maybe that receptor, when that neurotransmitter binds, lets something negative in. Remember we talked about that chloride over there? That's a negative charge. So if we let negative things in, that's going to go further from threshold. And it's going to be harder to send a signal. And if we make it harder for sending a signal, we call that inhibitory. So an inhibitory postsynaptic potential. So we're talking after a synapse here. We're going to make it harder. Now there's two ways to make it harder. You could let something negative in, or that receptor, when that little molecule comes and binds to it, could let positive out. And if you let potassium out a little too early, again, it moves you further from threshold and it makes it harder to send a signal. That's a lot of physiology in one video. So hopefully it kind of helped to see it just all kind of mapped out as one story. But the basic idea, gated ion channels open, you get a flow of charges, you add those charges up, doing summation. If you get to threshold, you create a signal. That signal is called an action potential. That goes down the axon to the end where you're storing your chemical message. Chemical message gets released into that space where it can bind to a receptor. If you want the next cell to respond, then you're going to stimulate it by letting positives in. If you don't want the next cell to respond, if you want to make sure it's getting turned off, then when you bind to receptors, you're going to get it further from threshold by letting negative in or positive out. Hopefully it was helpful. Thanks for watching.